can go over C. I actually put a slide on here because I knew y'all would ask me about that. So what exocytosis and endocytosis have in common are that a vesicle's involved. And what you want to think of it is basically, here's the cell membrane. Does anybody remember what a cell membrane is made of? It's phospholipids. Basically, a vesicle is a tiny little miniature cell membrane that's used to move things in or out of the cell. So that's a cell membrane. This little thing, or this little thing, either inside or outside of the cell, is a vesicle. So it's made of phospholipids. If you're doing exocytosis, let's say you've got a little protein, little triangle that's leaving. As it goes out, it kind of buds off, just like a budding, and it takes that piece of cell membrane with it and goes along its way. That's exocytosis. So it gets wrapped up in a little piece of cell membrane, and then it leaves.
So is it more important to just know like exo leave and endo?
That'd be the start, definitely. So remember, exo means what? Exit. So it's going out. Endo, going in. So the opposite. So you've got your little protein coming in, budding off, and it's coming in a vesicle. And then there's 3 parts. Or let me turn this light off. So there's 3 kinds of endocytosis. They all involve a vesicle coming in. But the main difference is phagocytosis. That's if you're eating like a whole bacteria or a virus. It's like done by immune cells. Pinocytosis is basically just drinking a little bit of the liquid around. So it's non-specific. You just bring in, 1 milliliter of the liquid in a vesicle. And then receptor-mediated is involved. You have receptors, they bind to something, they bring it in. I've got a little movie too.
The substances taken in by...
All right, I guess we can listen to them.
Phobic plasma membrane. Many single-cell eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use 3 major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as pinocytosis.
So you see I just brought in some of the liquid from outside. So they call it cell drinking. It's another word for pinocytosis.
Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein cladrine coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell.
So basically you've got a receptor that has a certain shape, and if something binds to it that has the shape that fits it, the cell brings it in a vesicle like that. Well, they just did one. This is a low-density lipoprotein, type of cholesterol in the body, LDL, like the bad cholesterol. Another example, that's one way that viruses get into your cells, is they'll bind to a receptor that's there for something else and trick the cell. Yeah, like bring me in, I'm a piece of food, but really I'm a deadly virus, and then it's too late. So those are those three. Anything else? I gave you just 3 things to learn, but they were all pretty complicated. Yeah, so B, basically simple diffusion, facilitated, and active. I think the best way to show that's with a table, probably. All right, so type. Right. Protein. Run out of room here. All right, so we've got comparing all 3 of these. All 3 of these are ways for the cell to move things in or out. So the first one is simple. Do y'all think there's a protein involved? Yes or no? No protein channels necessary. What about direction? High concentration to low or low concentration to high? So high to low. And does it take energy? No. What about facilitated diffusion? Protein? Direction? Still high to low. Energy? No. What about active? Channel? Direction? It technically could be both, but we'll just stick with low to high, because that's the difference. What about energy? All right. So that's the basics for those. We're going to talk more today about how you decide. So like if I give you a scenario, you'll have to decide which of those would be the answer, but not for your quiz. All right, so since it's the day after the Super Bowl, and maybe not everyone got my email, we're not taking a quiz today, but at least you're ready. But you still prepared for class, which is good. Should be ready. I can't actually bear to watch this. It's too disturbing and weird, but you can watch it.
I could have put a warning on there.
Did you click on it already? It's pretty bad. Were you scarred for life?
Yes.
Don't tell them what it is. Okay, well, it's up to y'all to find out. So disclaimer for today. Did you see how short the study guide is? It's still the hardest thing in this unit. People have the most trouble with it. So that's why it's short. So we're going to spend the whole time on it. Your first job is with your, maybe just in pairs, it'll work. I want you to draw a piece of the cell membrane on your table. Label it hydrophobic, hydrophilic, and I don't want you to use any books or anything. Just try what you can. See what you can remember. And we're talking about like a zoomed in piece of it, basically.
I could draw a cell membrane.
Oh, it'd probably be easier. Let's say you're zooming in on a part of it. Like, you know, zoomed in here. What would it look like zoomed in? Y'all can each do it. Here, you can be in charge of the green one.
No, I'll trade you.
All right, so what do you think a phospholipid looks like? It's got a head and two tails, each one.
So like that.
Yeah, exactly. So that'd be like one phospholipid. So think about if you had two layers of them, what it would look like. All right, that's good. So that's one phospholipid. So I want to see 10 on each side, like that.
So do it like
Oh, but each one gets its own head,
Oh.
right? So one phospholipid has a head and two tails like that.
Does it matter where the tail is?
So far it doesn't.
Okay.
All right, so if you're still stuck a little bit, let's go ahead and sort of review the answer to that one. Most people got it, but we'll go ahead and think about it. Sorry. All right, so here's your cell membrane. We're zooming in just on one little part of it. I like to draw mine beautifully like this. So of course, these tails, are they hydrophobic or hydrophilic? If they're hydrophobic. Hydrophobic. And then what's this head up here? What about down here? So just to orient ourselves, let's say this is the inside of the cell, this is outside. What else did you have to draw?
The transmembrane protein.
Okay. So transmembrane, what does trans tell you?
It goes all across.
Goes across. So if you're drawing a transmembrane protein, you could just draw any shape that goes from one side of the membrane to the other. Really common transmembrane proteins are these channels that are going to let things through. Because if you're going to let something through, you probably have to be on both sides, right, to open the gate. Makes sense? Peripheral is just on one side. So let's put a green one on there. Some shape on one side would be peripheral. They can be on both. Most of them are on the outside, though. Not all of them.
And they're like attached to the phospholipids, right?
Yeah, they can be attached to the phospholipids, or they could be attached to a transmembrane protein. But they're attached somehow to stay there. So that's that one, if you're stuck. So one thing that'll be important in a little bit, in order for something to get through here without help, it has to go past these heads and these tails. Just remember that for a moment. All right, what is the main function of the cell membrane, do you think? Now, this one, see, this is what everyone always wants to say, because every time you see a drawing, it looks like that's what's making it a circle, right? But the structure actually comes from the cytoskeleton inside the cell. So if you think about, this is just a phospholipid. Phospholipid's like a piece of soap. So our soap bubble's very strong, like barely at all, right? Same thing with a cell membrane. Very easily, they move very easily around. You could squish them. They don't really give you structure. That's the job of the cytoskeleton, which is stuff inside the cell. So really common misconception there. And to prevent dehydration, as you'll see in a moment, water is one of the things that can go straight through that cell membrane. The cell can't really control it. It just goes in, it goes out. And that's called osmosis, which is one of the things we're working on today. All right, so what can go straight through? Small polar things, which means they're slightly charged, but not very, very charged, not fully charged. Things like water is small and polar, carbon dioxide. Also, something that's small and nonpolar, or uncharged, completely uncharged, like O2. Your steroid hormones are also small and nonpolar. And that's because, do you think nonpolar things, are they hydrophobic or hydrophilic? They're hydrophobic. And so these tails here, they're hydrophobic. So they let things that are like them through. So that's the small nonpolar. Small polar, they're small enough and they're not all that charged, so they can sneak through as well.
How does that work though, like when you have something that's hydrophobic, but it's in a water environment, like the extracellular, how does it work?
So it's not really dissolved, it's just kind of moving along with it.
Okay.
So if you think about, if you put some oil in, a few drops of oil in water, and you just like poured it through something, the oil would stay together, but it'd still be moving along with the water.
It just won't freeze all.
Right. Mm-hmm. But once it gets to here, it sees these tails and it very easily passes through. Anything too large can't fit. That's pretty obvious. Now the confusing one is the small charged molecules. They are so hydrophilic that they're too attracted to the water and they'll never go through these tails. Mainly because they're so attracted to the water, they stay up above or they stay inside because they're fully charged, very, very attracted to water. So it's not just size, because ions, how many atoms is one Na+? That's one atom. So it's very small, but it's charged, so it can't go through the membrane by itself. Well, if it's plus, it has one less electron. And if it's negative, it has one extra electron. So we'll get back to all those. Let's talk about osmosis a little bit. I'm just going to match these up. All right, so which one is diffusion? So diffusion is D. So the diffusion is the movement of any molecule from higher to lower. Doesn't matter what kind of molecule it is. What's osmosis? So E. So osmosis is the diffusion of water across the cell membrane. That's osmosis. Gets its own word. What about isotonic? Isotonic is C. Hypotonic. So hypotonic is B. So that's bad. And then hypertonic is A. So what do you think 100% like pure water would be? Hypo, hyper, or iso? Pure water? It is hypotonic, so 100% water. And that is because inside of the cell it has stuff dissolved in it, right? It's not 100% water, it's got salts or glucose, everything else. So if you put in a cell in a hypotonic, or sorry, in 100% water, water is going to go into the cell because there's more water outside. Too bad.
Hyper.
If you have a solution of 100% water, it's hypotonic. If you have a solution that's, I don't know, 50% salt, it'd be hypertonic. And the best way that, I didn't come up with this, one of my students a couple years ago did. The first thing you do is figure out which way water is going to go based on comparing the inside and the outside. Because water goes from where there's more solutes to where there's less to even it out. And so they said, where do hyper children need to go? Outside, right? Send them outside. So their way to remember it was, if water is going outside the cell, that's hypertonic. And we're going to practice these two. It's a little confusing. So water moves from high solute to low solute. Or sorry, high solute to low solute. Which is the same thing as saying water at a high concentration to water where it's low. So for example, if you've got, where am I going to put all this? All right. And this is a really good one because it actually shows you. All right. So basically, you could imagine, left versus right side. One side could be inside the cell, one could be outside, like the solution it's floating in. So right now they're equal, right? Now if you add in some kind of solute, which is this green one, take a look at what happens.
Here we go. I need you in the bathroom.
So what do you see happening with those green those green urea molecules? So they attached to the water, and since water is bouncing back and forth, these waters that attach to the urea, they can't move anymore. They're kind of attached. So that means which side has more free water? It's the left. So which way do you think water is going to move? It's going to move to the right.
So then would it be hypertonic?
Well, it would depend which side of was the cell, right? Because they didn't tell you,
If they were moving, consider moving. Oh, okay.
right? Because you could say the left's on the inside, right? We'll practice it with the cell in a little bit. But basically, water's moving to where there's higher solutes.
Is there like a name for that property?
Well, the movement is osmosis. But I'm not sure what exactly property that would be. But the whole process is osmosis. So if you put a cell in a solution that's not equal, water moves, the whole thing is called osmosis. All right, so try this one on your own here. Let's say you've got this situation. Blue's water, yellow's sodium ions. This is inside the cell, and this is a beaker I'm putting the cell into. So I want to know which way water is going to go. Is it going to go into that cell or out of the cell? Let's give you a minute. Just try it all on your own. And we'll go over it. So let's go ahead and show you how to do this one. So the first step you do is find out the concentration of the solute in both places. So how would you figure out inside the cell? Concentration of sodium.
Pair up the water molecule with the salt.
You could think of it that way. Or you could just do it 2 over, right? So it's 8. Well, you could do it either way. I usually do it the total number of molecules. As long as you do it the same way on both sides, it wouldn't matter. But there's 2 sodium, there's 8 total molecules, right? 6 water, 2 sodium. So what percentage is that? Okay, so we have 25% Na plus. What about outside or in the beaker? I definitely meant to make that 2 over 10, but I can't count. All right, so which side has more solutes, inside or the beaker? So which way is the water going to go? Right, so the water is going to go from here. And that'll eventually even it out. Right, if you start taking water from this side, put it over here, maybe you'd get to 20% on both sides.
Do you have to do it that way every time?
What do you mean you have to do that way every time?
So do you? Like, can you just look at it?
I would advise not just looking at it, just to make sure. Sometimes it won't be that obvious. If it was a picture problem, you could, like you were saying, just take out the water molecules that are attached to the sodium or something. Think of it that way. Perhaps you could do it that way. I'd probably do it this way, but. All right, we're going to practice that some more in a second. First, let's talk about our cells. So if you put, for example, red blood cells, if you put them in a hypertonic solution, where does the water go? It's going out, so the cells shrivel up. If you put them in a hypotonic solution, does the water go in or out? In, cells would burst. This is why when you give, most of the time, what kind of IV are you giving somebody? Saline, do you know what percentage? Well, it depends if they're dehydrated or not, but if they're not dehydrated, it's 0.9%. And that's because that's what the inside of the cells are. So you don't want those cells to shrivel up or burst, so you give them an isotonic solution. If they're dehydrated, they get more hypotonic to even that out. Less salt. If they're too hydrated, which is much less common, they get a hypertonic solution. Right. So which of these is true of a cell put into pure water? better know which ones you think it is. Definitely the way to do it is figure out which way water's going to go first, and then say which kind of solution it is. That's usually the order of steps I would do it in. If you try to skip to the second step, you might confuse yourself. All right, so I've got 3 practice problems for you. All right, so for A there, inside is 1%, outside is 5%. Which way is water going to go?
Out.
It's going to go out, which makes it a hypertonic solution. And you know, because water goes to the higher solute area. What about B? Inside is 1%, outside is 0.5%. It's going inside, because now the higher solute's inside the cell, so it's hypotonic, water going in. And for C, what do you think? It is isotonic. If you have multiple solutes, you just add them together. So you have 6% inside, 6% outside, which means that's isotonic. Water has no net movement. All right. Next up, if you turn your worksheet over, you get to make your own problem. So I want you to work in pairs on this one, come up with 2-word problems for somebody else to solve. Fold it, put the answer on the bottom. All right, so basically just to recap, all you have to remember is, which way does water go?
To the higher concentration.
It goes to higher solute. So figure out where's the higher solute is. Water goes that way. And then if it's going out, it's what? Hypertonic. It's going in. Hypo. If it's not moving, it's iso. So that's the steps you want to do in your brain. Now what happens to people on the test? It's not that they're dumb or they can't figure this out. What do you think happens? You start to get nervous, you rush, and you try to skip to step 3, right? You'll like look at the picture and be like, that looks like more water and it's going to go that way. Instead of just taking it step by step, which I know you could do. Because those steps are easy if you just take them. So just don't rush. You won't mess it up. All right, so the second main thing, part of this chapter, is going back to simple diffusion, facilitated diffusion, active transport. This is what we're going to work on probably for the rest of the time. All right, so basically take osmosis out of your head. That's only if water's moving back and forth. If you're moving anything else, then we have to do simple diffusion, facilitated, active. That's kind of the way we're going. The first thing is which, like we mentioned before, what can go through the cell membrane without any help, without a hole. So you've got water, gases, small uncharged things, or something that's small and partially charged. And then things that can't, anything that's too large, and anything that's charged and small. It'll depend. So that's where we get to in a second. All right, so wait. So why can't ions get through the cell membrane? All right, we got 13 answers. Anybody else? All right, so I would say G is the best answer there.
So what was it before you put G?
Wasn't really right.
A would have been.
Yeah.
A was the right answer.
So basically they're hydrophilic, so because they're hydrophilic, those tails repel them. They stay attracted to the water outside. And this is the number one thing people always say, they're too big. They're the smallest thing. Ions are tiny, but they're charged, that's why they can't get through. The tails are hydrophobic, but the ions are hydrophilic, so they don't match.
Right? It's supposed to be neutral. No, an atom. It's supposed to be neutral. It's supposed to be balanced, right?
Well-
That's what makes it an ion, is that it's
Right, it's unbalanced.
unbalanced.
All right, so let's think about a cell. What kind of categories of things does a cell want to bring in? So proteins, nutrients, sugars, yeah, maybe enzymes. What do you think a cell wants to send out? A waste would be an example. What else? Well, lysosomes might send something out. So what about protein signals, maybe, like hormones? Signals to cells that are around? Ribosomes do not, but ribosomes make things that go out. And so the whole point of cell transport is for the cell to be able to do those things. Bring the nutrients in, send waste, send signals out. That's why it's doing simple diffusion, facilitated active transport. And of course, y'all had that table from earlier. So simple diffusion, no energy, only works if you're going from high to low. And most importantly, that only works for molecules that can go straight through the cell membrane. So those, small nonpolar things, small polar things. So like the gases, the water, steroids that we mentioned earlier. All right, now since this is simple diffusion, is the cell in control of which way it goes? No. It's not, right? It's just going to go from high to low, whatever side it's on. So if I ask you, set up a scenario and say which direction is going to go, this is how you'd solve it. So let's say oxygen. So 10% oxygen inside, 5% outside. So you're putting a cell in here. Which way is the oxygen going to go? So it's just going to go from high to low. So it's going to go out of the cell. What if I put, instead of oxygen, what if this was calcium ions? Which way are the calcium ions going to go?
Well, they can't move.
So the ions can't go through the cell membrane, so they're not going to move. So unless we're talking about water moving, you wouldn't say hypo, hyper. That's just a totally separate thing. So these are my kind of steps. Can the molecule move through the cell membrane?
No.
You say no, you stop. The molecule's not moving. If it can, figure out the concentration inside, concentration outside, and then the solute's going to move to the lower location.
then how did it move out?
Because
Back to the oxygen.
Because it wasn't charged, because it can go.
Yeah.
down the membrane.
Let's go back to the
But
oxygen.
inside-
This one. So it is
How does it move out if the 5% is smaller?
because it's not osmosis. So now there's more oxygen bouncing around inside the cell, and there's less bouncing around outside.
So it's like opposite.
So it's kind of opposite. Let's take a look at a movie. Oh no. The website's been discontinued. Oh well.
I can't click on this when I open one.
You have to go to slideshow mode to click on them. Otherwise, you have to copy and paste them. If you want a slideshow mode, you can click on those. And we are going to, part of that physio lab thing is going to be practicing with this. Let's get some more practice. All right. How about this one? So this is confusing, right? Because it's basically opposite of what you were just doing with osmosis. Because now we're talking about the solute moving, and the solute, there's more molecules inside than outside, so it moves out, the solute itself. So unless the question says something about osmosis or hypotonic, then we're talking about the solutes moving. So we're going to do a flow chart in a little bit. It'll help with that. And also those words, hypotonic, hypertonic, that only applies if water's moving. So you wouldn't be using those words at all. Just confuse you. All right. So facilitated, as we mentioned before, no energy. It works for molecules moving down their concentration gradient, which just means high to low. We had up here in the beginning. However, this is for things that can't get through the membrane, like the ions. So they need help of a protein. So for example, if you were doing this calcium 1 before, let's say there is a little calcium channel. Now the calcium can leave the cell and go into the solution because there's a special hole for it that opens up.
Is that like a transmembrane?
That is a transmembrane protein, yeah. Sure, so all the channels have a specific shape that'll only let either one or two molecules through. So like a calcium ion, you can think it's got like a triangle, and this channel has a little triangle thing that catches it. This one, the same thing is shut down. Okay, all right. I don't know what that was about. So here's your cell membrane. You can see your phospholipids, hydrophobic, hydrophilic, and these are like the channels. And things are moving much faster in real life. And so those are glucose, that's sodium. All right. So which side has more glucose, outside or inside?
Outside.
Definitely the outside. Glucose is too big to go through by itself. So if it tried to, it would just bounce off. It's too big, can't fit through. But there's the transport proteins that have a shape that'll grab the glucose and let it through. Like so, won't make me speed it up. And so here, If you look at the calcium, it doesn't have the right shape, so it doesn't fit to go through. It would need a different channel to be there.
What happens, like, when the concentration equals out? Like, do the proteins, do those channels close, or it just is constantly continuing because your cells are constantly using it? So.
Well, it would depend if it became equal. Most of these channels can work both directions. So you would just have one leaving, one going, one leaving, one coming in, once it got to the equilibrium. The way these were drawn, they're not gated. Once we get to muscles and neurons, we will talk about like the gated ones though, that are like controlled. But for now, just, they're open all the time. All right, and then for active, this takes energy. It takes a protein, and it's basically moving something from a low concentration to a high concentration. It's like you're moving water uphill. Maybe water's not a good example. You're moving anything uphill. It takes energy to do that, right? But like putting a rock on the top of a hill and letting it go, that doesn't take energy. But putting it up there does. And that's what active transport's doing. So you do need some kind of protein to do this for you. And it can move things from low to high. All right, which is this one? So you can see this little green molecule here. It's at a low concentration inside. It's some kind of waste product. But the cell wants to move it out because it's a waste. So this little star thing that comes in represents ATP or energy. And it uses that energy to push it out. Like so. All right. So let's make a little flow chart here. So another one of those multi-step processes to figure out question. You don't need to draw it. We'll do it on here together. So basically, for this kind of flow chart, you're looking for yes or no questions to figure out a problem. Go to Steve, so maybe not. So let's say you have a problem on a test like this. Calcium, 5% inside, 2% outside. And then let's say the cell all right, so the way these problems work, we'll tell you some certain molecule, concentration inside and outside, and which way the cell wants to move it, inside or outside. And then to figure out whether it's active, osmosis, facilitated, or simple, that's where you'd use the flow chart. So what kind of question could you ask to decide whether or not it's osmosis we're talking about?
Is it involved water?
Okay. So how about, is water moving? Question one. If the answer is yes, then what do we have? Okay, so we know it's osmosis. So we kind of know where to go. If the answer is no, we still got these other three to choose between. How do you know if it's active transport? Okay, but does that one say anything about energy over here? You're right, if I say it uses energy, you'd know. But I'm probably not going to say that.
Cross the membrane by itself?
That's probably going to be our next question, but that's not going to help us with active transport. Which direction does active do?
low to high.
And I heard low to high. Somebody. All right, so if you say it is going from low to high, what's the answer then?
Active transfer.
So you're at active. Because that's the only one up there that goes from low to high. So that's active. And if it's no, we've still got two more to decide between, right? Facilitated and simple. And somebody already told me the question. Okay. How do you know if it can cross by itself? Is it charged, or what's the other category of things that can't? There you go. So if you say yes, is it going to be simple or facilitated?
Facilitated.
Which means it needs help, right? So this is the facilitated. And if no, simple. All right, so the way you'd use it, we've got this one over here, this problem. Calcium 5% inside, 2% outside. Cell wants to move it outside. Personally, that's too many words and stuff in my head to really figure it out. So I would draw myself a picture. So calcium, 5%. Calcium, 2%. And the cell wants to move it outside. To me, this is a lot easier to figure out than just reading it. So if I read it, I'd draw a picture. If it was a picture, you just stick with it. All right, so then we look at this. Is it talking about water moving? So we're over here. Is it going from low to high? It's going from 5 to 2, so that's a no. Is it charged or large?
Yes.
So you go facilitated. So you know the answer is facilitated. All right, let's do some more practice. So try this one on your own. We've got 3 scenarios. Use this flow chart, and I want to know what the answer is for each. I would probably draw a picture for each of them, just to help your brain out.